Hello, welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture 16, where I will discuss the late steps of isoquinoline alkaloid biosynthetic pathways. Particular focus will be given only on the benzyl isoquinoline alkaloids, and then we will look into the cell and tissue localization of these alkaloids. And so, this is here you will find some of the latest discoveries and which led to the completion of the entire pathway, and that is very important. So, that from there metabolic engineering approaches can be undertaken, leading to changes in the pharmaceutically important alkaloids, both in the composition as well as in the contents. Okay, let me go to the topic concepts to be covered. So, first one is the benzyl isoquinoline alkaloids biosynthesis. As I said that late steps will be discussed now, and then cell and tissue localization, and then if time permits we will talk little bit of metabolic engineering, otherwise we will discuss in the next class. So, in the previous class what I have discussed is how this pathway originates. It basically originates from tyrosine and then it forms norcoclaudin, and then through a series of methyl transferase and hydroxylase, finally it forms reticulin, and from reticulin the pathway moves in different direction. One route which goes towards berberine alkaloid, protopine alkaloid and benzophenanthridine alkaloids. So, that is through the reaction catalyzed by berberine bridge enzyme BBE, leading to the formation of scauleriin, and then scauleriin further converted into these alkaloids through multiple step reactions. Whereas, in the right side what we have seen that reticulin salutaridinol is formed, and then from thebaine, and then we see there are two routes, and both the routes ultimately contributing to the formation of morphine. So, now this part we are going to see in little bit more detail. Reticulin, I have also said that it is the chemical chameleon. So, because that is the base molecule, and from that molecule different changes in the functional groups and modification of the alkaloid scaffold structures, leading to formation of different alkaloids in different plants. This is just to remind you. So, I just go to the board now, and I will draw from reticulin onwards. Reticulin, this reticulin S will be converted into reticulin R through two step processor, and then from this reticulin what we see the formation of salutaridin, salutaridine. This salutaridin will be converted into salutarinidol, salutaridinol, and this subsequently converted into salutaridinol 7O acetate, and that ultimately leads to the formation of thebaine. Now, I will put the arrows So, here activities of two enzymes are very important. One is SAL-R, which converts salutaridine to salutaridinol. So, this is salutaridine reductase, and the second one is SAL-AT, which is salutaridinol acetyl transferase, leading to the formation of salutaridinol 7-acetate. So, earlier work basically deciphered these steps, but much later scientists, when they use the molecular tools, they try to characterize these enzymes, and next what they try to do, they wanted to study these steps more. One particular interesting case I would like to discuss here, that one group of scientists, they try to block this step using RNAi approach. Okay. So, their aim is basically that if you block the salutaridinol acetyl transferase, then acetate formation will be suppressed, leading to the formation of salutaridinol. But what result was obtained was this, that it's not only enhanced the accumulation of salutaridinol, but also salutaridine. So, that means as a result of RNAi blockage, both these products are getting enhanced. So, there is no reason that this RNAi block will affect the salutaridine. So, and subsequently there is another group, they also achieved the same results, and from there they have come to some suggestion that both these enzymes are working in a common enzyme complex. So, what is called in the metabolome complex. So, that is, if one is affected, that will affect the other one as well. So, that means when SAL-AT function is impaired, that will affect the SAL-R function, and that is why salutaridine is getting accumulated at an enhanced amount. Okay. So, from this reticulin also the one route goes to berberine through berberine bridge enzyme, that leading to the formation of other alkaloids like berberine, sanguinarine and all other. There is another route which also contributes to other alkaloids, non-narcotic alkaloids, we will talk about this later. One is called laudanine, another is called laudanosine. So, these are all conversion possible. Now, let us see what is going to happen after thibane. As I have shown you in the previous slide, that thibane, from there are two pathway exists. Okay, now I am going to talk about that. So, thibane. One pathway from thibane, it makes neopenone, this is unstable intermediate, and then it moves further and makes codinone. And from codinone it makes codeine, and from codeine it makes morphine. Similarly, from the other route, thivane makes oripavine, and from oripavine it makes morphinone, and for morphinone ultimately it makes morphine. Now, so the enzyme responsible for these conversions are this CODM, that is COR, which is convert corinone to corin, COR corinone reductase, which converts corinone to corin and morphinone to morphine. Now, let me tell you little bit more about this two pathways. So, the left one, this pathway is operating in the Munich variety of opium poppy, whereas the right side one, Tasmanian variety of opium poppy. These two geographically different varieties, that means located into different geographical areas. Basically, what has been discovered by the scientists, that they evolved two different pathways. There are common enzymes, but ultimately it forms the final product morphine, but it differs in steps. Okay. Neopenone is basically an intermediate, and this conversion to neopenone to chorionin is basically a spontaneous reaction. Okay. Now, at this stage nothing much was known about the enzymatic reactions which convert corin to morphine or oripavine to morphinone. So, based on the structures, what scientists predicted, that there are likely to be demethylase enzymes which is playing important role. So, in other words, if I write here maybe a demethylase. So, here there will be a demethylase, here there will be a demethylase, and also the other reactions are also demethylase, but I will just put it in a different color shade. For example, and okay. So, this was the status until 2004, and then finally, this was resolved in 2008 onwards. So, let us now look into these aspects. Okay, let me before I come to these aspects, let me come to this discovery. So, here let me take the pen. So, a mutant discovered in 2004, basically it was reported in the famous journal Nature, that morphine pathway block in top 1 poppies. So, why and what was the rationale for doing this work? So, this is a mutant, top 1 is a mutant, but this mutant basically it is a not natural mutant. So, this was discovered, or this happened as a result of chemical mutagenesis. So, the laboratory, they took seeds of poppy and then they treated with mutagens, and then after that they have done vigorous skinning, and with this ultimately they have come up with discovery of one mutant which is called top 1. Now, question is that why they undertook such study? The reason is this, that apart from this morphine and codeine, there is also need for other morphine and alkaloids of the pathway, or in other words that scientists they determine that they will come up with a top not top with a opium poppy plant which will not produce morphine. Why? Because morphine can be acetylated, and that leads to the formation of notorious drug heroin. And so, this is called illicit. So, these are illicit drug. So, they want to have licit drug, that is opium poppy which will not produce morphine, so that it cannot be used further to make heroin. So, that is one of the objectives. Other objectives is basically to try to accumulate some of the intermediates of the morphine pathway in higher amount if the morphine is blocked, because those are also used for different products including thebaine. So, what I said that the top 1 mutant discovered, and what they found basically in the top 1 mutant, there is the blockage here. That means, the top 1 mutant when they analyze, they found that it accumulates high quantity of thebaine, but not morphine, not codeine. That means what? That means the route from thebaine to codenone via nepionine is blocked. So, and not only that, this route is also blocked, that is from oripavin to morphinone. So, how they have confirmed that? When you see the control, this is yellow, this one is thin layer chromatographic plates. So, here basically they grow these mutant plants, they allowed them to produce the fruits, and this is the capsule, and then from there they isolated the latex, and then they ran the TLC, and here this TOCM, T stands for thebaine, O stands for oripavin, C stands for codeine, M stands for morphine. In the control, what you find that there is a thebaine is present, codeine is present, morphine is present, that means the wild type. Okay. Whereas, in the top 1, what you found is basically a lot of thebaine and less amount of oripavin, but there was no trace of codeine and morphine. That means, the downstream pathway is blocked, and this third one is basically the standard, so that you can identify. Okay. So, and this is what is the HPLC analysis. The bottom one is basically the mutant, that is the top 1 mutant, and the upper one is the wild type. So, in the wild type, what you find? You find morphine, okay. You find codeine, morphine in higher amount. So, that is why most of the codeine got converted into morphine and little bit of thebaine. Whereas, in the top 1 mutant, you do not find any morphine. So, no morphine. Instead, what you find? You find higher level of oripavin and higher level of thebaine. Okay. So, that is what is shown in the HPLC traces of alkaloid profiles. The upper one is the control parent, which what I have written the wild type, and the bottom one is the mutant, and top 1 trace lacks any codeine and morphine, it is not there, and TLC results I have analyzed. So, thebaine and oripavin accumulate as the dominant alkaloid in the mutant, at the expense of morphine and codeine, which are evident in the parental phenotype. So, this is very clear, and this is basically an outstanding achievement, and that is why this paper published in Nature. Why outstanding achievement? Because we found what scientists found, that it is not producing morphine and codeine. That means that the pathway is blocked in the mutant. So, there must be an active enzyme which converts thebaine to neopenone, what I have shown in this earlier drawing, that thebaine to neopenone. So, that let me use a different pen. Thebaine to codeine. So, this step there must be, at least this step there must be an enzyme. So, in the mutant this enzyme is blocked, and in the mutant the this enzyme perhaps is also blocked. That is why you see lot of oripavin and thebaine accumulation. So, if that is the case, then there must be active enzymes, and likely from this study they suggested that these are demethylase enzyme. Now, later with the use of functional genomics tool, scientists finally identified these steps. So, these are the two previously undetected enzymes involved in morphine biosynthesis, which are unique among the plants, has been identified, and these are basically the deoxygenases. So, these were identified as T6-ODM, that is thebaine 6-O demethylase, which converts thebaine to neopenone, and neopenone is unstable. So, it converts actually thebaine to codenone. So, the reaction makes neopenone, but neopenone is unstable, it immediately converted into codenone. So, when you see the product, you will only see the codenone, and the other one T6-ODM converts oripavin to morphinone. So, this is one, and the second one what they have characterized, that is basically a CODM, that is codenone or codeine O demethylase, codeine O demethylase, whose job is basically to convert codenone to morphine and thebaine to oripavin. So, remember that this pathway is operating in the Munich variety, and this pathway is operating in the Tasmanian variety, and similar effect they found in both. So, what they found, that in both the varieties there indeed CODM exists. In one variety it converts thebaine to oripavin, particularly for the Tasmanian variety, whereas for the Munich variety CODM converts codenone to morphine. So, this is also a demethylase, and the Munich variety, the T6-ODM, that converts thebaine to codenone via neopenone, whereas in the Tasmanian variety it converts oripavin to morphinone. So, what I said, that these genes have been characterized. To confirm their function, what they have done, basically they made the construct in such a way that they try to block its function. So, one way of blocking this function, they have used a technique called virus induced gene silencing. So, through virus induced gene silencing method, they block the activity of T6-ODM and CODM in the respective varieties. So, if these are blocked, then what is going to happen? Then let us see. So, what is showing here in here A and B is basically the T6-ODM from the Munich variety and B is the T6-ODM from the Tasmanian variety. So, here what you see in the control, there is lot of, this is the substrate thibane. Okay. In the control means there is a lot of thibane, that is only the substrate, and when T6-ODM enzyme is active, this is just showing the enzyme activity. So, which the substrate is being utilized and you see the formation of the product. So, this product is nothing but the codinone. Similarly, the bottom one is for the Tasmanian variety, where the T6-ODM, here is the substrate is the oripavin, this is the oripavin, the substrate. The control one is that there is no enzyme, and T6-ODM where the enzymatic reaction is there. So, the substrate is getting depleted, as a result of product formation is there. So, this is morphinone. This is the enzyme HPLC, this is basically LCMS reaction, profile of the LCMS chromatogram rather, not reaction, LCMS chromatogram, which is showing the product formation. This I am telling at this moment, then I will come to the result, what happens as a result of this blockage. So, here in the similarly in the right side, so they have used CODM. This CODM is basically from the Munich variety, and this CODM is from the Tasmanian variety. So, here is also same, the substrate is codeine, codeine is converted into morphine. So, when CODM is active, all the substrate is utilized, morphine is formed in one Munich variety, whereas in Tasmanian variety it shows slightly different results, but it is fine. So, next we will discuss what happens as a result of gene silencing. So, the empty vector, I will only discuss only two lanes. Empty vector is basically the control. So, where you consider it as a control plant or wild type plant. So, here no gene function has been disrupted, and as a result of that, what you find? You find morphine, you find codeine, you find some oripavin and thibane. The second one is basically DIOX-A, that means you are totally blocking here at this step. When you block this step, then what is going to happen? All the downstream in this pathway, particularly the, say if we talk about the Munich variety. So, when you block here, all the downstream get affected, and as a result of that, what you see here only. So, if it is affected, then only thibane accumulates in high amount when you compare with the control, because in the control or wild type, that thibane got converted into the product, but here all the because it is the step next to the next to thibane, which converts thibane to codinone is blocked, all the subsequent step got affected. That is why you do not see any product formation or very limited or very very limited product formation here in this. So, almost it is nil. Same is true here when the alkaloid content was measured. So, what you see in case of empty vector, that is in the wild type, you see that it the morphine content, this is black, is morphine is detectable in substantial amount, even codeine is detectable, but thibane very less, because all thibane got converted into that. But now, if you see the DIOX-A, that is the one where CODM is blocked, what you see here is very less morphine, almost nil, codeine little bit, but a huge amount of thibane. Okay. So, what is shown in that thin layer chromatography, that is also confirmed by HPLC analysis, and the same was confirmed by gene expression using real time PCR. So, where in the control where it is called the empty vector. So, here all the genes T6ODM is active, DIOX2 is active and COR is also CODM is also active. Whereas, in this one, that is the where the T6ODM is blocked, all the expression got suppressed. So, that means, through this gene silencing, this is one tool, one can use RNA interference, one can use antisense suppression. So, virus induced gene silencing another way to rapidly check that. So, this is a much fast way of checking the output what one has achieved. So, and what has been found with this, that indeed they have characterized the right enzyme, and with this they finally elucidated the steps of morphine biosynthesis in both the cultivars or both the varieties. So, what we see here, what we have studied today is from reticulin S to reticulin R, then it moves in this direction, then it forms salutaridine. There is of course, a salutaridine synthase which I did not mention, then sal R is there, then sal AT, and from sal AT thibane, then thibane the one pathway moves towards codinone, codeine and morphine. So, which is the Munich variety, whereas the other one, that is the Tasmanian one, which moves in this direction, that is thibane to oripavine to morphinone and morphine. And now, I will show that you see that all these are well characterized. So, the we talked about T6ODM, COR already characterized before codenone reductase, and in this pathway CODM, T6ODM and COR, and the Munich variety this CODM. So, in other word CODM, T6ODM, COR, CODM, whereas in the Tasmanian variety CODM, T6ODM, COR. So, that means, the late steps of morphine biosynthesis has been characterized, and all the enzymes and genes are in hand of the scientist. So, with this they can do the manipulation of the pathway, and that leads to changes in the alkaloid profile, which is basically comes under the metabolic engineering, and that we will discuss in the next class. Before I end, I will take only few minutes to tell that these alkaloids, basically this is the anatomy of the tissue, the leaf tissue. So, where we find that so this is the vascular portion where you find the phloem and the xylem. So, the sieve element that and the companion cells. So, these are the main region where these alkaloids are accumulated in the green tissue as well as, but in the root. So, it is basically the pericycle, that is the main site of accumulation. So, what happens, these alkaloids, its biosynthesis is operating in the phloem. From the companion cells it comes to the sieve elements, and from there it moves to the latecifer, that is the specialized cells which accumulate latex, and there these alkaloids are safely stored. So, this is basically putative trafficking of this alkaloid pathway and the intermediates, okay. And this is the tissue specific localization of the enzymes and genes. So, this is very important, because if you want to study this properly, so you have to isolate these particular type of cells, and from there you can isolate the mRNA and do the subsequent work, and in fact scientists did such things. And nowadays there are techniques like single cell isolation of RNA, single cell isolation of metabolites, these are all possible. And this is again another way. So, basically what is showing that the yellow and red. So, there are 3 types of cells in the phloem. Yellow are adjacent to the SIB element, and the companion cell pairs which are red in color in the aerial organs. So, these are the places where the alkaloid accumulates, whereas in the root tissues it also accumulates in these areas, what I have said, it in the cortex as well as partly in the pericycle. So, alkaloid biosynthetic enzymes are assembled in the companion cells and subsequently translocated to the SIB elements, and then they transported into the latissifer for storage, which I have shown in the previous slide. So, and this is finally showing another way, a transmission electron microscope, what they are showing the location of the SIB element adjacent to the latissifer, so that alkaloids can freely move from here to here, and it moves from the companion cell to SIB elements to latissifer, and this is basically showing the bottom one is a plasmodesmatal connection. So, where these are the red are latissifers and the green are the SIB elements. From there the alkaloid moves from one to another, this to this direction, and this is a longitudinal section showing the places where these alkaloids are accumulated and move from one tissue to another. So, with this I end the class for today. Thank you very much.